University of Rwanda. So let me take this opportunity to thank the director of the Center of Excellence in Biodiversity and Resource Management, Professor Beth Caprin, to give me this opportunity to share this knowledge for this weekly seminar. So let's get started and welcome to this presentation. So let me share my screen. So I think you see the screen, right?
Yes.
Yeah. Okay. So our topic, we're exploring microbial diversity and ecological function of epiphytic and surface sediment biofilm community in shallow tropical lake. So we perform our experiments in Lake Rumira in Eastern Province in Bugesera. So what are biofilm? So biofilm are matrix enclosed microbial aggregates comprising bacteria, algae, fungi, and metazoa. So these biofilm, they are enclosed in the matrix. That matrix contains lipids, polysaccharide, extracellular DNA. Also, they have some connection inside the matrix where there is channels and voids, where those gaps inside the matrix, they are used by microbe to communicate using the process called column sensing. So those biofilm, they may grow on biological and non-biological surfaces in form of mucus, mats, biofilm, and flow in suspension. So they may grow on woods, plastics. So any surface, those biofilm may grow there where they perform certain function, also where they may cause some harm. The example is what you call biocorrosion, where some biofilm, they may cause a lust for some metals, and the lust may cause economic loss because some pipes may be damaged by the biofilm. So where can we find those biofilms? So they are everywhere. They may colonize the wastewater treatment plants. They may colonize the surface of plants. So you may find the biofilm everywhere. They may colonize any surface or substrata. So based on the attachment, we may classify the biofilm as epipelon or episalmon. They may be classified as mats or epiphytic biofilm. So epiphytic biofilm, they are those biofilm which grow on the surface of the plants where they perform certain function like water treatment or biogeochemical cycling. So how the biofilm can be formed? So there is different stage of biofilm formation. First, the biofilm, the few cell, maybe bacterial eukaryote, they attach to the surface. Then they start multiplying. So the first step is the attachment. The attachment may be reversible or irreversible. Then follow the maturation 1 and 2. And once the biofilm grow, they can burst, and some microbial community inside the biofilm, they may do what you call dispersal. Then they go to colonize the new area. So importance of biofilm macrophyte system. There are those biofilm colonizing the plants. So they may have physical law as well as chemical and biological law. The physical law, the macrophytes are they are economic engineer because they can shape the physical property of aquatic ecosystem. They reduce water flow velocity. They facilitate the sediment particle settling. They may regulate water chemistry as well as clear water status. While the chemical and biological law, those plants growing underwater, they may be ideal substrate. They may shelter those microbes growing on the surface. So also they are basic component of aquatic ecosystem, while they perform different function such as microbial structure, primary production, trophic interaction, and so on and so forth. So what pushes us to do a research on this epiphytic biofilm and as well as sediment biofilm? So most aquatic macrophyte biofilm study focus on planktonic micro. So they most study focus on those micro which grow single, not in colony, but furthermore, they study on the influence of microhabitats, seasonal shift on eukaryotic diversity and interaction in epiphytic biofilm, and surface sediment remain largely unexplored and poor understood. So also, moreover, most study came from Asia, North America, Europe, and Australia, while the African continent it may still lag behind in this regard. So most studies were studied in the West and East, but the African continent lag behind in this regard. Also, therefore, the potential of exploring biofilm in aquatic plants and surface sediment is very significant due to their role in ecological restoration projects as well as eutrophication and climate change mitigation. So, to the best of our knowledge, this is the first of its kind to provide a comprehensive exploration of the influence of microhabitats, season, environmental variables on bacteria and eukaryotic diversity, assembly process, interaction, as well as ecological function in epiphytic biofilm and surface sediments. So this is the sampling. We sampled we take our sample in the east in Bugesera. So we took some aquatic plants, sediment, and some water. The water we measured the physical parameter as well as chemical parameter, while for the plant and sediment, we analyze microbial community. So the plant we sampled were Ceratophyllum, demersum, and Eicchornia crassipes. So these are some of the physical parameters we measured. These are chemical one. So these are some of the DNA extraction were extracted in College of Science and Technology lab, and the DNA sample were shipped to China for PCR amplification, gel electrophoresis, and for higher throughput sequencing. The data analysis were analyzed using software like Origin Pro, SPSS, Kanoko 5, RStudio, Cytoscape, and GEFI. Some statistical data were compiled or were computed using the STAMP tool. Our result, the main concentration of the mean concentration of temperature, pH, DO, EC, TDS, and nitrate were significantly higher in the dry season compared to wet season. However, TP and TN, as well as ammonia, reveal a reverse trend. So this condition of increased nutrient pollution in Lake Rumira can be ascribed to inlet rivers, agriculture activity near the lake, as well as siltation in the tropical region. So these were the main influence of the increase of these chemical and physical parameters. So we explore the bacterial diversity on different plants and sediments. So there was no significant difference in the alpha diversity indices. However, OTU richness was generally higher on demersum when compared to crassipes and sediments. While we performed the PCA for the bacterial diversity, we found 52% of the total variance and revealed that the bacterial community dissimilarity was different among substrate but not among season. So this may be due to the connection between the water, sediment, and the plant, so which may cause this homogeneity of the diversity between this substrate. Also, we perform the bacterial composition among the microhabitats, where we found OTU number were higher in wet than dry season, and this value were more in demersum than sediment and crassipes in two season. Most dominant bacteria phyla were significantly different among aquatic plants and sediment in dry and wet season. So we find the abundance of actinobacteria, cyanobacteria, chlorophylla were significantly higher in demersum than crassipes, where proteobacteria was significantly higher in crassipes. So taken together, the discrepancy in bacterial dynamics across different microbial habitat and season was possibly due to the substratum type, season, and environmental filtering. So these were the substrate type, the season, and environmental filtering were thought to be the influencing factor for this microbial composition. So the first one were the bacteria community and diversity, but here is also the diversity of microeukaryotes. For microeukaryotes, Shannon diversity and OTU richness of microeukaryotes were significantly higher in sediment than those in epiphytic biofilm from demersum. Also, the PCOA for microeukaryote community explained about 35 of the total variance and was statistically different among substrate but not among season. So this result may be due to the nutrients concentration as well as the environmental filtering. For the composition of microeukaryotes, the relative abundance of Notifera, Chloroplastida, and Gastrotricha was significantly higher in demersum than crassipes and sediment. However, the relative abundance of Ascomycota was significantly higher in sediment than in aquatic plants. We perform what we call redundancy analysis, which show that water temperature, pH, DOTN, and EC were the most important abiotic factor closely related to microbial community on these aquatic plants. Furthermore, we perform the prediction of microbial interaction and function in epiphytic and surface sediment biofilm. So the co-occurrence network using GEFI reveal more complex interaction among microbes on sea demersum than E. crassipes and sediments. And those interactions include cross-feeding, parasitism, symbiosis, and predatism among organisms in biofilm. So function predictive analysis include degradation of organic pollutants, hydrolysis of polysaccharide, nutrient cycling, and energy production to be positive effect on lake ecosystem and the sustainability of the local activity. We saw that on the plant called sediments, they show the complexity of the network were higher compared to these plants. So the summary, bacterial alpha diversity indices were insignificant among the different hosts. Microeukaryote Shannon value were significantly higher in sediment than sediments. Substrate type shape the microbial diversity in epiphytic biofilm and sediments, while the RDA result indicate that pH, DO, temperature, TN, and EC were the environmental drivers of microbial community in epiphytic biofilm. Microbe on sediments show complex interaction than other substrates, while the ecological law through function predictive analysis included degradation of organic pollutants, hydrolysis of polysaccharide, nutrient cycling, energy production, which could have positive effect on lake ecosystem and the sustainability of local activity. So for the second study, we look on the law of epiphytic biofilm and suspended superficial sediment in water quality of lake in tropical agriculture area by looking the interaction between water quality and microbe. The first study were done on Rumira Lake, but this one were done on Chohoa's North Lake. This is background. Our study investigated the analysis of land. We wanted to see the effect of land use type and the water environmental change. Also, we investigated the fact driving the microbial community assembly. Moreover, the interaction and ecological function also were compared. This is the sampling in Chohoa Lake. These were the measurement of water quality we measured. These were the methodology, the DNA extraction, PCR, gel electrophoresis, and the bioinformatic tool we used. This show how the Lake Chohoa were polluted or were under the eutrophication, where there is inappropriate agriculture activity, water eutrophication, and encroachment in the Lake Chohoa buffer zone. So the population are practicing the agriculture near the lake, and there is inflow of this chemical in the lake, such as fertilizer and so on and so forth. Also, by doing the land use cover with using GIS, we found that the Lake Rumira has been affected by agriculture. You see this area, they were green, but as the time goes on, there were agriculture development in this area, which may cause some pollution in the lake. So the factors in the lake, including and nitrates, the parameter eutrophication the lake. By model and neutral model, stochastic microbial community assembly in epiphytic biofilm and surface sediments. So the mature epiphytic biofilm and regularly cycling water flow may be ascribed with the phenomenon that stochastic, the microbial community assembly in aquatic plant and surface sediment. So we saw that stochastic processes were dominant, and we thought that this were due to the mature epiphytic biofilm as well as the river inflow, which may increase the stochastic movement. So by doing the co-occurrence network, the stability and the complexity of network differ among substrate, being significantly higher in surface sediment than sea demersum and crassipes. So co-occurrence network analyzed at the phylum level. So the node in 3 network were specified in 16 microbial phyla. Among these phyla, Proteobacteria, Firmicutes, Actinobacteria, and so forth were more distributed, accounting for nearly 75% of the node. But Proteobacteria was the most dominant phylum in all microhabitat network. So the summary for this study, stochastic process dominated the microbial community assembly in epiphytic and superficial sediment biofilm. Also, the stability and complexity of network differ among substrate, being significantly higher in surface sediment than sea demersum and crassipes, while the phylum Proteobacteria, Firmicute, Rotifera, Gastrotricha, and the other one play key role in the microhabitat network. So these are recommendations. So the general public should be made aware of the significance of aquatic macrophyte and their biofilm in water purification process and treat caused by emerging pollutants in aquatic ecosystem. So environmental monitoring studies should be conducted to know the status of freshwater pollution. For example, various pollutants must be treated before releasing into the wetland, lake, and river. So there are several key areas where research is currently lacking. For example, including epiphytic biofilm to understand, maintain, and improve freshwater ecosystem health and integrity, global study on microbial biodiversity, assembly process interaction in biofilm on different aquatic plants. Thank you and welcome for the question.
Thank you so much. It was really fascinating to learn about a whole new facet of biodiversity. We really appreciate, and also the benefits to that community, the biofilms. So I can see that, let's see, there was a hand up. I don't know, maybe that was a mistake, but I want to invite people. You can put your hand up and unmute and ask a question, or put your comments into the chat. Questions for Benjamin about what he found in these lakes. Yeah, I guess while we're waiting for questions, I'm curious, you're getting a lot of thank yous in the chat, but I just wanted to ask you, Benjamin, do you think that there's ways to use what you found with these biofilms for indicators of freshwater ecosystem health or lake health? Like using it as an indicator or tool? And I guess the second part of that question is, we in the center, we have different researchers working with different taxon groups, and sometimes they're finding new species. They often are collaborating with international partners, and we have to send material out. I know I've talked with Professor Antoine is here in the meeting as well, and he's been with me when we've talked with RDB about the export permitting, and it's often an issue to get material out. So I was wondering what you think about the potential to be able to do the sequencing work in Rwanda, because I heard you say that you sent the DNA to China. So given that, you know, you're now kind of an expert in this area, what are your thoughts about how we can get up to speed to be able to do that part of the work here in Rwanda? So those are my two questions.
Okay, thank you, Prof. Beth. So actually, the epiphytic biofilm can be used as indicator of aquatic health, because, for example, if some pollutants enter in the system, they may cause some injury to this microbe. So by sequencing, you may see some function were hampered because of these pollutants. So if you enter at the molecular level, you may predict, or you may find, for example, if the richness is lower, so this contaminant may lower the richness, may lower the diversity. So also this epiphytic biofilm may act as indicator for aquatic system health. So for the second question, exactly, for this sequencing is very important, for example, for the study here in Rwanda, because nowadays, for the study to be accepted in this higher journal, you need at least look at the molecular level in this area. So it's possible. And for example, in China, the sequencing is very cheap. Like, for example, one sample, let me say, it's like 20,000 for the sequencing. And if you want, they analyze your sample, and they may put in the system, and you can access the first few file, and you can do further analysis. So I have some connection of the people there. For example, if there is someone going there, sometime it is possible to give him some sample, and he can go there and do sequencing. Then they can send some file online, and you can.
Yeah, thank you. That's helpful to know. We may get you involved in the discussions we're having around the importance of being able to share material. I'm not sure if you're frozen. Can you still hear us, Benjamin? Good. Yeah, we have a question from Placide. He's asking, do you think that the bottom sediments would have an influence on your results due to the lake's mix?
Okay, thank you, Prasid. So actually, for the sediment, the superficial sediments, I don't know if he asked the superficial sediment. If for our study, the superficial sediment, we find that they have a lot of the higher diversity compared to epiphytic plants, because these bacteria living in superficial sediment, they are rich in diversity and composition. So for our study, we found that sediment had a higher richness and diversity compared to the plants or water. So,
did that answer your question, Placide?
Thank you. Excuse me. So I was asking about, because I saw you've been analyzing different parameters, physical and chemical parameters, which also influenced your results in one way or another. So I was asking myself, the bottom sediment from the bottom of the lake, Kyohoa or another one. So if, for example, the lake may mix from surface to the bottom, so I was wondering, those sediment from bottom, have you ever thought if they can have an influence on your result, final result?
Okay, thank you, Placid. So for the physicochemical parameter, we measured only for water. For the sediment, we didn't measure the physicochemical parameter. Only the microbial diversity and the sequencing of sediment. So possibly this bottom sediment may have the influence because, as we know, the bottom sediment of the lake is like a sentinel or the sink of the chemicals, the pollutant, they go there, they sink down. So of course there is what you call sediment resuspension, where there is a self-pollution on the bottom, from the bottom of the lake. So there is the mixing of those sediments may influence the water quality. But for us, we didn't measure the physicochemical parameter of the sediment. So possibly this may have the influence on this bacterial community and the composition.
Okay, thank you.
I'm sorry, I was muted. So are there other, anybody else want to ask a question? You can unmute or you can put it into the chat. I don't see any other questions in the chat right now, but you're welcome to go ahead and unmute and ask a question. Everybody awake out there? Any questions you want to ask of our speaker? Oh, good. Franklin, welcome. Go ahead. Franklin's one of our PhD students in School of Science. Welcome, Franklin.
Thank you, Professor. I'm very pleased with presentation of Dr. Ben. It's really good, but I would like to know, according what he presented, because it is a really new topic for us, but I would like to know what may be the relationship between the biofilm as he exposed with plankton and all aquatic lives. For examples, as a kind of aquatic plants, even aquatic animals. What may be the relationship among biofilm and these living life which are inside water? Thank you.
Okay, thank you. So there was a relationship between the previous study showed that some aquatic biofilm have the relationship between with the planktonic microbe. For example, these plankton, once they fix on the surface and there is attachment, growth, maturation, and dispersal. So this plankton may migrate to the surface and form the biofilm. But on the planets, they found that 95% of the microbe exist as biofilm because it's the best way for the microbe to fight or to withstand different threats like high temperature, salinity, and other chemicals. So it's like a protection, a microbial cloth which protect those biofilms. So the plankton, once they go on the surface of the substratum, they may colonize that surface and become the biofilm and will be protected by the matrix. So, of course, the plant, the animal, on their surface, there is biofilm there. And those biofilm, they have the function for each organism living in the water or on the earth. So there is a relationship between planktonic life and biofilm life, because plankton may migrate, do some maturation and become biofilm. But studies show that the best life for microbe is by biofilm life, not by planktonic way.
Okay, thank you, doctor. But I would like to know again if there is a same, what am I say? If it's same in fresh water and salt water, like lakes or seas, the same process may be possible to those two different ecosystems.
Thank you. So I don't know if I get well, if in fresh water and seawater, this relationship is the same. So,
no, I'd like to know if the amount, if I can say amount, if or average of biofilm is the same in fresh water or seawater. They can be same.
So the amounts must not be the same, because this biofilm composition diversity, they vary depending on the environmental parameters. So, for example, in seawater, there's a quantity of salt, so it may influence the composition, the richness, and other factors. So it depends on the area where they are located.
Okay, thank you so much. Thank you, doctor. Thank you. I'm really satisfied. Thank you.
Great, thank you, Franklin. Let's see, there's a question from Augustin. He's asking, now we are facing the question of climate change. How do the biofilms and plankton help us to adapt? That's an interesting question. Thank you, very timely. Thanks, Augustin. Benjamin?
Thank you. So for this question of climate change, actually, there is, if I'm not wrong, there is no study about the climate change on epiphytic. For other biofilm, there is, but no study on the climate change on the epiphytic biofilm. For other, it may there may be a research, but for epiphytic biofilm, I didn't see any paper about it. And this is a new insight which people may think and work on it. But of course, the climate change may have the effect on this biofilm, because this, for example, like global warming may influence the microbial diversity and composition, of course. But I think there no work on the epiphytic. For other biofilm, there may be there, but for epiphytic biofilm, there is no work on this one.
Good, thank you. Yeah, that's very interesting. So, do we have any other questions, other comments from people here? Do you have plans for other research, Benjamin? Looking at other ecosystems?
Thank you, Prof. Thank you, Prof. Actually, when I was here in Rwanda in 2019, I took also the sample for hot spring, Bugarama and Kisenyi,
Wow.
Nyamyumba. So I did the sequencing. The paper is already finished, but I will send it in few weeks after the exam. So it is very interesting. And also we partnered with another guy, Dr. Ale. Also we test if we can see the antibiotic resistance gene in the mats growing in hot spring.
Wow.
Then so it's uninteresting. So the paper are already done. So it remains with submission, and I think it will be a good one.
Oh, fantastic. That's really exciting to hear. Yeah, because as you know, we're also working on that dwarf water lily, which is endemic to those hot springs. So that's super. Yeah, any other questions or comments for our speaker? Well, if not, I want to thank everybody so much for coming today. Thank you so much, Benjamin. That was really a wonderful talk, and we hope to invite you back to talk maybe about what you found in the hot springs. And
Appreciate it.
meanwhile, I just want to encourage others, if you have something you'd like to talk about in this seminar series, feel free to contact Vinan Zibaza and he can get you set up. So thank you, everyone, and have a great afternoon.
Thank you, Professor Beth
Bye.
and
Thank you.
Dr. Benjamin.
Thank you.
Thank you.
Okay,
Bye.
thank you. Bye.
Bye.
I